Good morning, everyone. Welcome to week number 3 of Bio 120. Let's see what we've got going on. So last week we learned about some chemistry, the chemistry of life. You learned about the importance of water. It acts as a solvent, is cohesive, adhesive, it absorbs heat, and that's the reason that it is found in cells. You also learned about the organic molecules, carbohydrates, which provide energy storage, and also structures in cells. Lipids, which are hydrophobic, which are water-fearing, and so they act as a barrier. The phospholipids act as a barrier between the outside environment and the inside of the cell in the form of the cell membrane. There are other functions of lipids. They're fats, oils, and waxes, and so they're used for energy storage, and mostly they repel water. Also, the proteins, most of the proteins are enzymes which run chemical reactions in cells, but some of them are structural, some of them are even hormones like insulin. And then we had the nucleic acids, which often serve as the genetic information in cells, which pass along the code for producing that particular organism into the next generation. And DNA contains the information for making all the proteins that a cell needs. Then you also learned about carbon and the fact that carbon can form 4 bonds with other atoms or molecules. And that's why it is the basis of organic molecules, because it can make very big, very complex molecules, and that's what is needed for life. Okay, so that was last week. This week, you're learning about classification, and this is important. You need to know the difference between prokaryotic cells and eukaryotic cells. Prokaryotic cells are all bacteria, all bacteria. So if you say prokaryote, you could just as well use the word bacteria. Prokaryotic cells are very simple. They have no organelles. There's not a nucleus. There are no endoplasmic reticulum, Golgi apparatuses, mitochondria, chloroplast, nothing like that in bacteria. They're extremely small cells. And so the DNA is not enclosed in a nucleus. It's just floating around in the cell. On the other hand, you have eukaryotic cells, and so all the rest of the organisms on the planet are eukaryotic cells besides bacteria. So that includes animals, plants, fungi, and the protists. And protists include everything from algae to an amoeba to paramecium. Oh, there's so many more. It has a very varied kingdom, the kingdom Protista. From huge organisms to unicellular organisms, but they're eukaryotic also. So that's classification. And also in the module overview, it gives you an idea of how classes are ranked. So you start with domains and then kingdoms, phylum, class, order, family, genus, species. And we talk a lot about species, but we also talk about kingdoms like the kingdom Animalia and that sort of thing. So what are these? These are groups where we group together organisms that have like characteristics. And so the kingdom, the domain eukaryote contains all the eukaryotes. And like I said, that includes animals, plants, fungi, and protists. And so that tells you that we all have something in common. Yes, we have things in common with a mushroom. And that's our cell structures. All right, so that's classification and cell structures. So eukaryotic cells have more complex cells, and they do have organelles, nuclei, mitochondria, chloroplast, if you're a photosynthesizer. The endoplasmic reticulum, Golgi apparatus, and there's lots more, vacuoles, etc., storage compartments of all different kinds. Okay, so the chapters to read this week. 20-1, 20 section 1, chapter 20, section 1. Read that, and chapter 4 in your OpenStax Biology 2nd edition, free textbook. So please use it. It's a good textbook. And since it has a search feature online, you can go directly to these chapters. You don't have to fiddle around with anything else. Read the Module 3 overview and watch the videos. You will need those. And they're very interesting. You know, I'm a microbiologist, so I love all this cell stuff. I'm just saying. I even have little toy microscopes hanging around everywhere. Um, so the cells are very interesting, but one of those, I think it's the article that you read, is all about the microbiome. And we just don't know very much yet about the microbiome. So what the heck is that? That's all the microscopic organisms that live in and on us. And if you think about it, we have all different types of ecosystems around and in our bodies. So you might have E. coli, which you do, and I do, and most animals do. We have E. coli bacteria in our large intestine. You're not going to find E. coli in your mouth. You're not going to find E. coli in the conjunctiva of your eyes. Okay, you're not going to find E. coli down in your lungs. So the microbiomes all around your body, from your skin all the way to the inside of you, like your large intestine, is very varied. And we don't understand all of this yet. This is a very hot topic in research right now in microbiology. What are these organisms that live in and on us in these different locations, and what is their purpose? How do they keep us healthy? Because most of them keep us healthy. They outcompete any pathogens, disease-causing microbes, that get into us. They keep them away. They even have all kinds of strategies to kill off pathogens. So, what's the purpose of these microbes, and how can we ensure that we keep them? Have you ever heard of probiotics? So if you take an antibiotic, antibiotics do this crazy thing. They kill bacteria. Well, sometimes they don't discriminate between the bad ones and the good ones. I mean, how does a little chemical, an antibiotic, know the difference? And so it just kills them. So what happens sometimes is that you get a bad case of diarrhea because all of your normal flora or your microbiota in your large intestine have been killed off by the antibiotic. It is always a good idea to take probiotics. And what are those? Those are replacement organisms to help you repopulate your large intestine. Whenever I'm on an antibiotic, which I have been a few times, not that many, but a few. Whenever I'm on one, though, I will go to CVS and I will look through the microbiotic capsule section of the drugstore, and I will pick the one with the longest list of microbes on it. That's what I do. If one has 5 and another one has 13, I'm picking the one with 13. Since we don't know exactly what combination of microbes is important, I'm going with the more the better, because something in there is going to take in your large intestine. Some people do prebiotics, and that is consuming microbes before you get sick, before anything's wrong with you. I don't follow that idea because your body and your microbiome is very well tuned. And so you don't need to be adding all that extra stuff if nothing's wrong with you. Makes sense, huh? Homeostasis. Remember that from week 1? Okay, so anyway, watch those videos. I'm going to post a video which is absolutely amazing. It's the Harvard Antibiotic Resistance and Bacteria Study. I will post that. It just takes a minute to watch, but it is absolutely amazing. It shows you how fast antibiotic resistance develops in bacteria. This is an 11-day study, and they used E. coli in a huge sheet of agar, which I don't know how in the world they kept that sterile, except for the E. coli. But anyway, and they introduce different amounts of the antibiotic into different sections of the agar. So please watch this. It's going to show you how fast antibiotic resistance happens. You think, and most people think, that, it takes decades. Okay, sometimes it takes decades for antibiotic resistance to happen in bacteria. But watch this video and see how fast these bacteria become antibiotic resistant. Okay, so what do you have to do this week? You have the discussion board postings. And I'll give you some clues about this. So one of your questions is, why do antibiotics, which are drugs that kill bacteria, not viruses, not anything else really. So why do antibiotics target bacterial cells and not ours? So that's a really important point. When you take antibiotics, you don't want them to kill you, even though they're killing the bacteria, right? That's really important. Otherwise, you don't have much of a market for your drugs. Anyway, yeah, so there's got to be a difference between bacterial cells and ours, where this chemical kills these little cells and not ours. It has everything to do with cell structures. Remember, our cells are eukaryotic cells. Bacterial cells are prokaryotic cells. There are major differences in our cells, and that's what antibiotics target. So you're going to be looking that up. All right? All right, and how does our use or excessive use or misuse of antibiotics increase the rate of antibiotic resistance? What is antibiotic resistance? People do not become antibiotic resistant. Bacteria do, right? We take antibiotics, but the antibiotics just get flushed out of our bodies eventually. So bacteria become antibiotic resistant. They develop strategies to get around any antibiotic that's in their environment. So how do they do this? Why do they do this? Why does this resistance happen? And how can we prevent it? That's all a big discussion now. It is one of the top 10 public health issues in the world today, antibiotic resistance, because we are running out of antibiotics. And this is true from some types of tuberculosis bacteria, gonorrhea bacteria, Staphylococcus aureus, which causes staph infections. You probably heard of MRSA before because it's been all in the news for at least the last decade. But some of these populations of MRSA, methicillin-resistant Staph aureus, are now becoming resistant to other antibiotics, including vancomycin, which is one of the last antibiotics that we have to kill these bacteria. Hot topic in the world of microbiology, in our world, because eventually we're going to run out of antibiotics. All right, in your response posts, you need to discuss the antibiotic use in livestock. So for many decades now, we have been giving antibiotics to chickens and turkeys and pigs and cows, etc., to help them all grow bigger or lay bigger eggs. And this is not in sick animals. This is in perfectly healthy animals. So why do we give antibiotics to these animals that serve as food animals for us? Why do we do this? And should we do this? And are there any pathogens, remember disease-causing microbes, in these animals that could easily transfer over into us? And then what if they're antibiotic resistant pathogens? Yeah, this is a problem. There have been a lot of movements to get rid of this in livestock. If you go to the grocery store now, you can find packages of chicken, for example, that say no antibiotics have ever been used in raising this chicken. Now, you're going to pay a hefty price for it. I know, it chokes me up when I have to go pay $11 for a packet of chicken to cook. But they're there. They are there. It's going to cost us more, I do believe, but we sure don't want to encourage antibiotic resistance, especially in our food animals. Okay, and then you're going to begin your final project. Remember to choose your topic. I talked about those topics last week in my video, so if you missed that, go back and watch at least the last part of the week 2 announcement video. Choose your topic from the list, or if you're waiting until the last minute to do this, email me with an idea for another project topic, and I will tell you whether you can use it or not. And if you just decide, well, you're going to choose it on your own and not ask me we may have a problem with this milestone 1 because it has to be a big enough topic for you to cover as a final project, but also condensed enough where you don't go talking about everything under the sun. Though climate change, global warming is one of the topics, and that is a huge one. So anyway, email me if you have an idea for a project topic other than any of them that are listed. All right, follow milestone 1, guidelines and rubric. This is the introduction section for your final project. And use the correct format. This is supposed to be an outline, which is a Word document, but you have to have some detail. I mean, it can't be just single words. It needs to be some detail about each one of the points in the outline. And label your sections. This is not an essay. No part of this final project is an essay, like your journal assignments, for example. This has sections, and I need to know the sections because otherwise sometimes I can't tell the difference. You just start writing and writing and writing, and I don't know which part is what you're considering, where this thing happens, whatever it is, or how it happens. Yeah, that's how confusing it gets. So you have to label the sections too. So let's take a look at some of the parts. Here is milestone 1 with the rubric. The rubric's attached. This is only the first page. So there are the choices that you have. If you don't like those, just ask me. This is the introduction section, which is describing this topic. So, of course, this is an outline format, but when it says describe, that means several sentences like a paragraph. And a paragraph is 3 sentences or more, okay? It's not just one word. Why is it significant or of interest to you, and why is it significant in the field of biology and to the world? And give some real-life examples. Also, the topic, where does this happen? So if you're talking about global warming, duh, it happens throughout the whole biosphere. It affects the whole biosphere. But if you're talking about eutrophication of a pond near you, what you would say is that this affects this particular pond, whatever the name of it is, the water ecosystem here, but also the plants surrounding that ecosystem. So you see what I mean? Does it affect an ecosystem, a particular one? Does it affect all ecosystems, which some topics do? Does it affect only city areas around the world, etc.? How does it happen? Describe how this process happens. So if you take eutrophication, for example, how does eutrophication happen? You get a decrease in the oxygen. Your water may heat up for a reason, like global warming for one, but also the seasons. What happens to the nutrients in the system? See, so you need to describe the process to me. And who? What are the species that are affected by this, and especially which ones benefit? So if you're talking about eutrophication, the algae that cause the algal blooms, they definitely benefit. Human beings benefit because we've been dumping fertilizer into the water, into the waterways that eventually get out to the ocean. And we fertilize those little old algae, and they bloom right away and create horrible toxins and cut off the oxygen supply for all the animals, including manatees. I think this last year we had the biggest manatee kill-off in Florida that we have ever seen, which is crazy, and that's because of algal blooms. But who benefits from the topic? Which industries benefit? If we talk about eutrophication and algal blooms along the coast of North Carolina, which is what I'm familiar with, there are lots and lots of hog farms. Excuse me. Hog farms right along the coast, and all that waste gets washed out into the ocean, and we have algal blooms. And so the hog farmers and the people who eat pork benefit from this. So that's the who section. If you have any questions whatsoever, I can't remember if I have another slide. Yeah, I do. If you have any questions whatsoever about your topic, how you're supposed to write this up, what information to include, please email me. I will be glad to talk with you about this. So my tip of the week is to put your assignments into the correct format. There's always a description about how this is supposed to look, whatever it is that you're doing. So for this milestone 1, this first part of your final project, create an outline. I described that. But have enough information where I know that you know what you're talking about and you're telling me what the answers are. For example, in that introduction, describe this thing that you want to talk about. Air pollution in New York City, or eutrophication of a small pond where you live, or global warming and climate change. No. So tell me about that. You're going to use this outline to create slides for your final project. So keep that in mind. Milestone 1 should be submitted as a Word document, double spacing. This is always important for me because it makes it easier for me to read. If you cram everything up into a little teeny paragraph with little teeny font, it makes it very hard for me to read, and that is not a good thing. That aggravates me because I have a lot of these to read, okay? So just saying, make it easy for me to read. All right, and the instructions tell you to use 12-point font, Times New Roman, all that, and 1-inch margins. Do you think that I'm actually going to take a ruler and measure margins? No, but don't make it a skinny little column or all the way to the edges. Make it easy to read for me, okay? Minimum of 1 page, I would say that's, whew, I don't know how you could get all this information into 1 page. Or 2, but remember you're also going to have references. Each time you turn in a milestone, you're going to use at least 3 references. So that can be a totally separate page, your reference page, okay? Use in-text citations. Give credit where credit is due. If you got the information from somewhere, give credit. Plus, this is not an opinion piece, okay? You have to support all your information with other information from outside. So this is why you do research in the Shapiro Library. You can also do research online. Make sure you use really good sources. I will check them. But use in-text citations because, like I said, this is not an opinion piece. This is you providing information about a topic. It can be your opinion, but you have to back it up with other research, with other information from other sources to give some credence to what you're saying, okay? So there's my name. I'm Dr. Sigmon, m.sigmon@snhu.edu. Let me know if you have any trouble this week, if you have any issues. I will be glad to help you work through it. And that being said, I hope that you have a wonderful week this week.